To our dear colleagues, distinguished researchers, and to all the participants of International Association for the Protection of European Dark Bee, a please good afternoon. Today I'm happy to speak about Apis mellifera mellifera, the Prikamskaya breed type honeybee breeding in Perm region of the Russian Federation. So this project is a compilation of all honeybee research experience in Perm region, and it's being carried out by researchers from various institutions. So in the second slide, I will present several of them, including co-authors. Introduce briefly some of our bee breeding experts. Professor Arkady Ivanovich Shurakov has begun comprehensive survey on honeybees in the Perm region in the late 1970s. The honeybee research was developed in 2015 at the Department of Zoology of Perm State Pedagogical University under the supervision of Professor Alexander Vasilievich Petukov and Professor Mikhail Kimovich Simankov. So the research was carried out at the Perm State Agro-Technological University. The expansion of the Perm region across Russia has provided this ideal environment for the survival, natural selection, and growth of Apis mellifera honeybee colonies. So all these mentioned experts in the Perm region who have published and did outstanding research on Apis mellifera mellifera honeybees. So as given on the map, the Perm region is located in the east part of Russia and the western slope of the Middle Ural Mountains. So here apiculture in Perm region has been practiced since the 12th century. On the next, the summarization of the scientific and practical aspects of using beekeeping products. So those are interesting worldwide that have been highlighted in several scientific papers. The apiculture products are consumed by humans, their qualities are used in traditional medicine, and the pollination by bees is necessary for agriculture. For all these interests to be realized, there must be healthy honeybees and well adapted to their habitat. The breeding Apis mellifera honeybee resources for the preservation of nature and the honeybee product development as a complement in seem to be of general interest to beekeepers and to the researchers. Honey production during an active season is one of the most significant metrics in evaluating honeybee colonies, so the following slide has related information. Russia's whole land is split into 8 federal districts, each of which is represented by a different color as illustrated on figure 2. Olga Federal District, which is outlined in green, includes the Perm region. All the production is gradually rising in all regions. The Perm region, unlike the Far East, does not lead in honeybee colony productivity for a variety of reasons. Please note that the data given for the productivity of honeybee colony in Russia for the last 2 decades consisted all Apis mellifera species. Apis mellifera honeybees are the native population in the Perm region, and the general characteristics of bees are presented on the next slide. Pointed out the general characteristics of Apis mellifera mellifera subspecies, and these apply to all Apis mellifera honeybees regardless of their habitat. It should be emphasized that owing to environmental conditions, honeybees have an annual natural selection of lengthy winters and short abundant honey harvest. As a result of these findings, the features of Apis mellifera mellifera bees compared to those of other honeybee subspecies in examined in Russia. For the behavioral include features such as light honey print, aggressiveness, and swarming. If proper climatic conditions are present, the queen's high lay egg activity determines the likelihood of rapidly expanding the number of bees within a month. The honeybees can easily collect the honey thanks to their strong nectar dispersion. It is also worth noting that honeybees may survive a lengthy break time while still developing under favorable conditions. The genetic honeybees can be displayed in morphometric characteristics or phenotype. At approximate weight of 110 mg, honeybees have large body size and black color. Short proboscis, high cubital index, and average tarsal index are all important features to examine for. So these criteria are used to assess honeybee breed features in Russia. The following slide I will present the registered honeybees of Apis mellifera. Apis mellifera accounts for approximately 60% of all naturally occurring and domesticated bees in Russia. So the figure 4 illustrates one of the bee apiaries of Apis mellifera. The honeybees are kept in the hives, so those bees discriminate between native populations that are geographically separated and have distinct environments. They form the foundation of honeybee breeding. The breed Bashkirsky, Tatarsky, and Burziansky produced utilizing purebred breed method on the basis of Bashkir, Tatarsky, and Burziansky populations. Orlovsky breed type of honeybees was developed as a result of mating 11 different populations of Apis mellifera, so they are imported from natural habitat. And the breeding type Prioksky was developed by the staff of the Beekeeping Research Institute on the basis of crossbreeding of the Apis mellifera and Apis caucasica breeds. Patents have already been filed for this species and kinds, so for the Prikamskaya breed type, the procedure of collecting documents just begun, and this breed was developed from the Prikamskaya population in Perm region. Each breed and bred type were obtained on the special apiaries by natural conditions in which the breed type was developed and produced. On the next slide, apiaries marked on the map was demonstrated. Figure 5 shows 7 locations where Apis mellifera honeybees can be found and certified pedigree apiaries. The Perm region is home to 3 of them. The following slide on figure 6 shows the position of the Perm region. Perm region forms part of the northern frontiers for the natural distribution of honeybees, spanning about 645 kilometers north to south and 420 kilometers west to east. The distance between the northern Krasno-Vishnyovsky and central Usinskaya Honeybee Research Center is around 359 kilometers. The distance between the main and southern is approximately 81 kilometers. The distance between the northern and southern apiaries for Honeybee Research Center in the Perm region is around 359 kilometers, resulting in various honey collection times and types of honey plants, and also with variation of interim duration. As a result, Apis mellifera Prikamskaya breed type is divided into northern and southern breed types. Northern Apis mellifera honeybees was moved to the apiaries of the northern part of the Perm region, and it was sold by the company Perm base. Since 1998, the former honeybee breeding station near Yasipovskaya has been kept to preserve southern Apis mellifera honeybees. The company Tentorium is the founder of honeybee breeding station Parazol on the preservation of Apis mellifera honeybees, which is the southern Prikamskaya breed type. More than half of the Perm region covers by forests of dark coniferous zones, the middle and the south. Plants in the central taiga forest differ from those in the southern region, which illustrated on figure 7. The breed of northern Apis mellifera honeybees settled in mountain woods. The mountains provide extra seclusion for the honeybees. The breed of southern Apis mellifera honeybees settled in the southern taiga and mixed forest, where honey collection is the longest and overwintering is shorter, and living conditions are more suitable for bees than in the north. The following slide shows the overall plan for studying honeybees by researchers from the Perm region. The honeybee research intended to evaluate local honeybees based on morphological and ecological characteristics in order to protect the colony in the wild. They also created a database of morphometric and behavioral characteristics of honeybees from various ecological zones in the Perm region, such as environmental conditions, number of beekeepers, total number of honeybees, apiaries, and their locations were described. And these findings from a honeybee living habitat, as well as the honey resource assessment, should all be taken into consideration. The major melliferous plants of the Perm area are shown on the slide. The various plant species such as linden, rani, honeysuckle, willow, and rose hips have the largest proportion. Lindens, elms, and maples are all common melliferous plants in the southern taiga. They don't grow in the central taiga, and linden is only found as bushes there. The intensity of nectar production varies throughout the season, as illustrated on figure 9 on the next slide. The figure 9 presented the characteristics of nectar production for each natural zone during the period of honeybees' active time. Perm region, the major maximum honey harvest occurs in July, and this is the only time of year when honeybee collect honey. So nectar release in May and June is highly dependent on weather conditions, and it can only be used to supplement honey production when the bee colony is unable to produce enough honey. The study on the honeybee breed trait was carried out according to the characteristics of behavioral and morphometric analysis. data is presented on the next slide. The honeybee morphometric analysis in region revealed that 52% of bees have Apis mellifera characteristics, 40% of honeybees have characteristics that do not fully correspond to Apis mellifera characteristics, and 8% of bees belong to the subspecies of Apis carpatica. Because of their capacity to adapt to many environments and their strong vigor, however, most beekeepers prefer Apis mellifera honeybees. Southern subspecies of honeybees have higher death rate. Therefore, they must be imported every year. The morphometric characteristics used to determine which subspecies and species of honeybees belong to each other are listed on the table on the next slide. The morphometric standard for Apis mellifera honeybees based on 5 criteria, and it was adopted based on long-term morphometric research on honeybees in Russia. According to Rutner, a low level of variation indicates that the trait size is stable. Crossbreeding, on the other hand, increases the diversity of morphometric characteristics by more than 20%. Morphological identification of the honeybee colonies was carried out based on features and indices such as length of proboscis, length and width of the wing, length and width of the target 4, cubital and dorsal index. The length of proboscis of the northern and southern honeybees was the minimum and within the standard of the Apis mellifera honeybees. Similarly, the average of width of flowering within the standard. However, the length of flowering of Prikamskaya breed type honeybees did not exceed the standard limit of Apis mellifera, but the average of southern honeybees was lower than the standard. The cubital index of southern honeybees in the northern honeybees corresponded to the Apis mellifera standard. Therefore, the north and south Prikamskaya breed types of honeybees differ by very stable characteristics, allowing us to differentiate them within the Perm region. Honeybee habitat conditions and related behavioral characteristics differ the north and south of the Perm region as shown in the following slide. The reason for the comparison analysis of honeybee samples from the northern and southern Apis mellifera honey breed types were the climatic differences in habitat conditions, which resulted in differences in honeybee activity. The period of overwintering and active periods for honeybees in the south of the region is 6 months, then in the north is about 5 months, and overwintering lasts for 7 months, creating more severe conditions for survival of honeybees. For its common features for Apis mellifera mellifera honeybees in the Perm region are that during the active season, colonies grow quickly. The high and productive lay activity of the queen determines the possibility of rapidly expanding the number of bees in June, less often in May, and over the course of a month depending on weather conditions. July is the month for the highest productivity. These honeybees makes an excellent use of the late May honey harvest for the gathering honey sufficient for the existence of bee colony in all other months with an inconsistent supporting honey harvest. If the main honey harvest lasts only months, the overwintering period lasts on average 215 days, and it's marked by the inability of honeybees to leave the hive for 6 months in the south and 7 months in the north, as well as the need for honey in the hive throughout the winter and early spring. In August, queen's capacity to lay eggs declines, and the honeybee colony lacks brood on September. Apis mellifera provides extended broodless protection against varroa mites because meteorological factors restrict the growth cycles of both the honeybees and their parasites. These characteristics are also evident in bees' increased illness resistance. The next figure, which is figure 11, depicts the genetic study of Apis mellifera Prikamskaya breed type honeybees using the genetic molecular DNA markers. In figure depicts the location of the intergenic locus as mellifera. The genetic purity of honeybees from both the south and the north of Perm region may be seen on the histogram of the amount of introgression of genes from thousands of species and population of Apis mellifera. The genetic distance between the honeybees in the Perm region south and north may be shown in dendrogram of genetic relatedness among local populations of Apis mellifera. The dendrogram is based on the analysis of microsatellite polymorphic loci. Therefore, the genetic study distinguishes the northern and southern Prikamskaya breed types of honeybees, allowing us to differentiate them within the Perm region. On the next slide, in the morphometric and genetic investigation, the physiological characterization of Apis mellifera honeybees was investigated. A comparative study of Apis mellifera Prikamskaya breed type with the imported Apis mellifera carpatica breed type was performed to discover distinct changes among honeybees. The most important attribute for honeybee survival in difficult overwintering conditions was determined to be dominant winter hardiness traits. For the physiological assessment, we noticed that during the overwintering, the catalase activity was decreased by January and increased by the end of the period. The catalase activity prevents the metabolic pathways from oxidative stress by elimination of radicals. We found that the average catalase activity of honeybees had narrow margins in January during the state of deep overwintering, whereas the phosphate reflects the energy requirements for the honeybee colony. It was shown that the energy depends on the location in the honeybee club. We also determined end of overwintering by enzyme activity, where the several Apis mellifera carpatica honeybees showed increased enzyme activity that reflects the increased metabolism before brood rearing. In other honeybees, the metabolism did not differ before overwinter season. At the same time, in Apis mellifera honeybees, overwintering metabolism is determined by enzymes activity. Therefore, it was the genetic need to end the overwintering earlier in Apis mellifera carpatica honeybee than in Apis mellifera honeybees. So this all was regardless of the environmental influences. When considered with other factors, these indicators lead to the death of Apis mellifera carpatica honeybees throughout overwintering season in the Perm region. We also noticed that the temperature of maximum hypothermia of honeybees differ during the overwintering season. So there is annual and very often meetings of beekeepers, research, and officials are being held to in the natural population of honeybees. On the next slide, the procedures for breeding honeybees in the breeding apiaries was described. The identification by phenotype, or more accurately by behavior, is the most accessible to beekeepers. These are the usual tasks for each beekeeper in the apiary that completes once a year. On the right side of the slide, the most frequent factors for evaluating honeybee colonies are given. Throughout the procedure, beekeepers transport honeybees to a mate queen in a cage. Within 12 days, cages with queen and honeybees were successfully delivered. On average, it takes about 3 to 4 days. Beekeepers were contacted to exchange information on the progress of honeybee colonies formed from the queen supply. On the following slide, I will talk about the reserves in the Perm region. Named Vyshyarsky in the north and Malinovoy Hutor in the south were established to safeguard honeybees and maintain the population's unique gene pool of Apis mellifera honeybees. The establishment of the Perm Reserve Malinovoy Hutor in 2002 to safeguard honeybees and maintain the natural gene pool of the population of Apis mellifera was one of the group's great achievements. Moving to the next slide, the Beekeepers Association are also established to facilitate contact between beekeepers and the breeders. The locations with the highest number of honeybee colonies are indicated in dark in figure 14a. It is clear that beekeeping is more established in the Perm region south area. One issue is that despite the death of the southern honeybees, some beekeepers import queen bees from south annually. In the nearby apiaries, this resulted in uncontrolled crossbreeding of local Apis mellifera honeybees. The Perm region, on the other hand, is one of the few sites in the world where the original gene pool of Apis mellifera honeybees has been conserved. Moving to the next slide. Russian land is divided into 8 federal districts, each of which is marked in different color on the diagram. The Volga Federal District, which is outlined in green, includes the Perm region. In the Volga District, the number of honeybee colonies is increasing and beekeeping is becoming more popular among individual families. On the last slide, highlight several suggestions and future prospects for the development of the honeybee breeding in Perm region of Russian Federation. There should be an exchange of experiences between Russian and international honeybee breeders in order to promote the optimal queen rearing techniques. The second is the state protection zones encompassing Apis mellifera honeybee breeding apiaries should be established in order to protect the gene pool from any imported or random honeybees. The third is the development of direct correspondence with beekeepers will be set up to exchange of information and assisting in the management of honeybee colonies from received queens. And the last but not least, the increase of the number is also important in Apis mellifera pure honeybee colonies in private apiaries in the Perm region to prevent the loss of the gene pool due to uncontrolled mating with southern bees. So this was all today. Hope the information presented here was very useful, and we really appreciate that you took time to be here and listen to our presentation. Next time, thank you for your attention. Отлично.